Okay, I will start. Today we are talking about the tooth development. This is our first topic in the block about teeth. Teeth are a major component of the oral cavity. Children have 10 deciduous or primary teeth in each jaw, 20 of them. And during a period of years, usually at beginning about of age of 6 years and ending at about age 12 to 13, deciduous teeth are gradually replaced by 16 permanent or secondary teeth in each jaw. Babies have incisor teeth, canine teeth, and 2 pairs of molar teeth on both jaws. They do not have premolar teeth. Adults have all types of teeth, including 2 pairs of incisor teeth on both jaws, pairs of canine teeth on both jaws, 2 pairs of premolar, and 3 pairs of molar teeth. We should discuss in general the tooth structure. So the tooth has 2 main parts: the crown, that is visible above the gum, and inside the gum is the root of the tooth. Between the crown and the root is the cervix, or a neck of a tooth, and we have the clinical and anatomical cervix. The clinical cervix is the site of attachment of the gum epithelium to a tooth, and the anatomical cervix or anatomical neck is a boundary between the enamel and the cementum of the tooth. Enamel is the most hard tissue in our organism, or some of the researchers do not consider the enamel as a tissue because it doesn't contain the cells. This is the mineralized extracellular matrix produced by the cells, enameloblasts, that are not yet alive in the adult mature tooth. They die when they end the production of enamel, and the root of the tooth is covered with the cementum. So the boundary between the enamel and cementum is the anatomical neck of the tooth, anatomical cervix. Inside a tooth we have the pulp chamber, filled with soft tissue, loose connective tissue with a lot of blood vessels and nerves inside it. Also, in the peripheral layer of the pulp, there are located the cell bodies of the cells that produce the dentin that covers the pulp in the area of both the crown and the root of the tooth. So dentin covers the pulp. The cells of dentin, dentinoblasts, are located outside the dentin in the peripheral layer of the pulp, but their processes are located inside the dentin. So these radial lines are the locations of the dentinoblasts, inside of the processes of dentinoblasts, which cell bodies are located in the pulp. And the tooth is attached to the bone, alveolar bone, with the help of periodontal ligament. This is the dense regular connective tissue. Here in this diagram you can see these radial lines. These are the directions of the bundles of collagen fibers that are connected with one end to the cementum of the tooth and with the second end to the bone tissue of the alveolar socket for a tooth. On the apical side of the tooth root, we have the apical foramen. This is the hole that connects the pulp chamber with the outside tissues. And through this apical foramen, the blood vessels and nerves enter inside the pulp chamber. Now, about the tooth development. In the process of tooth development, we have these 4 stages of tooth development, or odontogenesis. First stage, the stage of dental lamina, and next the dental bud. Dental lamina and dental bud. Second, enamel organ, that includes the stages of cap and abel. The third stage, the stage of histogenesis of dental tissues. Histogenesis means production of tissues. And final one, root formation and teething, the eruption of a tooth. The formation of milk teeth starts from 2nd month before birth, and teething is at the 6th, 7th month after birth. And the permanent teeth are formed 4 months before birth up to 3 years of life, and are erupted at the age of 6-7 years of life of a child. What do these stages mean? Stages of tooth development. Stage 1 includes stage of dental lamina and dental bud. This is dental bud. This is the stage of tooth germ initiation. Second stage, stage of enamel organ, that includes the cap stage and abel stage. The cap stage is tooth germ morphogenesis, which means the formation of enamel organ, dental sac, and dental papilla. The bale stage is the stage of tooth germ differentiation or cytodifferentiation, in which the cells start to differentiate into progenitors of particular dental tissues, into odontoblasts and enameloblasts and others. Also, residual dental plate that connects the enamel organ to the epithelium of oral cavity disintegrates. And also, during this stage of bale, the future shape of the crown is determined, whether we will have the incisor or a molar tooth, for example. During stage of histogenesis, the amelogenesis, dentinogenesis is occurred, which means the production of the tissues of the tooth, enamel, dentin, loose connective tissue of the pulp of the teeth, and other. And finally, root formation and teething, eruption of a tooth. The root is formed just before the eruption of the tooth. And during this stage, the cementogenesis occurs and development of periodontal ligament. Now we are talking more closely about all these stages. Stage 1, formation of dental lamina. During the onset of the tooth development, the oral epithelium forms the invagination that goes around the jaw in the horseshoe shape look, and the 2 invaginations are formed, the dental lamina and the vestibular plate. This is the cross section, how it looks like. Dental lamina is posterior here, and the vestibular plate is anterior. And this is the front view from the lip of a child. Soon the vestibular plate the cells of vestibular plate in its center will die with apoptosis, and the buccal labial sulcus will form that separates the gum from the lips and cheeks. So this will be the area between the gums and lips and or cheeks. And the dental lamina then will develop into the dental bud. At the site of each future tooth, there is a further proliferation of cells that arise from the dental lamina, resulting in production of a round cellular bud-like structure that projects into the underlying mesenchyme. This outgrowth, referred to as a bud stage, represents the early enamel organ. 10 buds are formed in each jaw. These will be future 20 teeth of a baby. The mesenchyme is actually the neuromesenchyme because it arises from the neural crest. And during the next stages of development, the epithelium tissue induces the differentiation of mesenchyme, and vice versa. Mesenchyme tissue induces the differentiation of the epithelial structures of enamel organ. In this picture, we can see the cross-section of the face part of the fetus of some animal. In the center, we have the tongue. This is the oral cavity, upper jaw, lower jaw, and a few dental germs, including the upper teeth in the stage of dental lamina. So this is the epithelium of the oral cavity that invaginates into the layer of mesenchyme. So this is dental lamina in this part also, and the two teeth inside the lower jaw are at the stage of enamel organs. Also in this picture, this is the oral cavity, the tongue, upper jaw, lower jaw, and the developing teeth. This one in the lower jaw at the stage of the dental lamina, and this one at the next stage, the enamel organ. The formation of the enamel organ, the first stage of enamel organ is the cap stage. Developing tooth in the cap stage shows its connection with the oral epithelium. This is the enamel organ, which connects to the oral epithelium. The enamel organ consists of single layer of cuboidal cells that form the outer enamel epithelium, here and here also. The inner enamel epithelium that consists of columnar cells that will in future differentiate into enameloblasts, and the layer of cells adjacent to the inner enamel epithelium that form the stratum intermedium. The remainder of the structure is occupied by the stellate reticulum or enamel pulp. So here is the stratum intermedium and here is the stellate pulp. All these parts of the enamel organs are constructed of the epithelial cells. All these cells are epithelial of the ectodermal origin. The mesenchyme of the dental papilla has proliferated and pushed into the enamel organ in this direction, and also here. At this stage, the forming tooth is surrounded by the condensed mesenchyme called dental sac, which gives rise to the periodontal structures. Condensed means that here the cells are more closely located, closely to each other, if we compare with these mesenchyme cells. So here you can see the structures. Red one is the enamel organ that will give rise to enamel of the tooth and cuticle of the tooth that will disappear during the eruption of the tooth. Yellow one is dental papilla of mesenchyme cells. It will give rise to the dental pulp and dentin of the mature tooth. And green one, the dental sac, that will give rise to the cementum and periodontal ligament of the tooth. Here are the structures that can be seen on this photograph. Dental lamina connects the oral epithelium with the enamel organ. In the enamel organ, we can find the outer epithelium, stellate reticulum, or the pulp of the enamel organ, intermediate enamel epithelium, and inner enamel epithelium lying on the basement membrane, whitish line here. The enamel organ is surrounded with a dental sac, and here we can see the dental papilla. Next stage, formation of the bell stage, bell-like stage. In this stage, the connection with the oral epithelium is almost cut off in this area. And the enamel organ consists of a narrow line of outer enamel epithelium, inner enamel epithelium formed by future ameloblasts, several condensed layers of cells that form stratum intermedium or intermediate layer, and the widely spaced stellate reticulum. At this stage, the epithelial cells of the stellate reticulum or the pulp of the enamel organ change their shape from the multiform polygonal shape that was seen in the previous stage into the stellate shape. And now they resemble the mesenchyme cells, but they are not mesenchyme, they are epithelial cells. Between the cells, the water with dissolved nutrients is accumulating. The dental papilla is now deeply invaginated against the enamel organ. At high magnification, the part of the enamel organ, here is the dental lamina connecting the enamel organ with the oral epithelium. Here the cells of the outer epithelium, here also pulp of the enamel organ or stellate reticulum, the stellate-looking cells with thin processes with a lot of extracellular matrix. This is the water with the solvents that will give the nutrition in the future for ameloblasts. Intermediate layer and the inner epithelium located upon the basement membrane, under which we can see the cells of the dental papilla. In our microphotograph, the tooth development early stage, we can see the enamel organ that is connected with the dental lamina with the oral epithelium. The enamel organ consists of the neck of the enamel organ, outer epithelium, inner epithelium on the basement membrane, here the intermediate epithelium, stellate reticulum, and these are all the structures of enamel organ. Enamel organ is surrounded with dental sac, and here is the invagination of the dental papilla, consisting both dental papilla and dental sac of the condensed mesenchyme cells. Here we can see the non-condensed mesenchyme cells, and these are bony trabeculae, bony spicules, that are forming future bone of the alveolar bone tissue, the bone socket for the developing tooth. And here is the part of the Merkel cartilage. Now, in this diagram, you can see the bell stage of the enamel organ, also consisting of outer outer, inner, intermediate epithelium and pulp of the enamel organ. Also, dental papilla and dental sac are visible here. The neural crest-derived pre-odontoblasts lined up within the bell adjacent to the inner enamel epithelium become columnar and now will have the epithelial-like appearance. So these are the cells of the peripheral part of the dental papilla just beneath the basement membrane of the inner epithelium, and these cells of dental papilla of mesenchyme origin will soon develop into pre-odontoblasts that will be then developed into odontoblasts that will produce the dentin of the tooth. The inner enamel epithelium consists of cells that will develop into the ameloblasts that will produce enamel of the tooth. Along with the cells of the stratum intermedium, they will be responsible for enamel production. At the early stage, just before dentinogenesis and amelogenesis, the dental lamina disintegrates, leaving the developing tooth primordium detached from its site of origin. Here we still see the dental lamina, but soon the cells of the dental lamina will die and it will be disintegrated, but some residuals of these cells can still be found inside the tissue and form the islets of Masse. Now we should talk about the cytodifferentiation of pre-odontoblast and pre-ameloblasts. We can look closer to this area where you can see the connection of the cells of the inner enamel epithelium located on the basement membrane, a white line here, and the underlying cells of the peripheral part of the dental papilla. So these are the cells of the inner layer of the enamel organ that are now differentiating into pre-enameloblasts. They are epithelial cells that are located on the basement membrane. At their apical part, they are connected with the cells of the intermediate layer of the enamel organ. Under the basement membrane, you can see the cells of peripheral layer of the dental papilla. These cells will soon differentiate into the pre-odontoblasts. And here, the inner part of the dental papilla, consisting of the mesenchyme cells. The differentiation of the mesenchymal dental papilla occurs simultaneously with the differentiation of the epithelial enamel organ. It starts from the site of the future apical part, side of the tooth crown, and continues downwards towards the future root of the tooth. Reciprocal induction between the cells of the inner enamel epithelium and the cells of the peripheral layer of the dental papilla make them to differentiate into the pre-enameloblast and pre-odontoblast respectively. The cells of the inner enamel epithelium differentiate into pre-enameloblast and release some biological active substances that pass through the basement membrane of the inner epithelium and act on the cells of the dental papilla located just beneath the basement membrane. As a result, these peripheral mesenchyme cells epithelium and the cells of dental papilla receive the nutrition and oxygen through diffusion from the blood vessels, blood capillaries located in the dental papilla. The cells of the inner epithelium receive this nutrition via diffusion through the basement membrane. Next, the pre-odontoblasts then differentiate into the odontoblasts or dentinoblasts that start producing pre-dentin towards the basement membrane of the pre-enamelloblasts. Pre-dentin consists of organic matrix, like it is in the process of formation of bone tissue. When mineralized, the pre-dentin turns into hard dentin that blocks the diffusion of nutrients to the pre-enamelloblast from the blood capillaries of the dental papilla. It makes the pre-enamelloblast to differentiate into enamelloblasts or amelloblasts. They change their polarity. First, they had basement membrane that separated them from the pre-odontoblasts. Now, this basement membrane disintegrates, and the basal pole will be at the site where the pre-enamelloblast contact with the cells of the intermediate layer of the enamel organ. The enamelloblasts now receive nutrition from the interstitial fluid between the cells of the pulp of the enamel organ. As you remember, the cells of the pulp of the enamel organ receive their stellate shape, star-like shape, because they accumulate a lot of water with the nutrients dissolved in it, and now they perform the function of give the nutrients to the enameloblasts, pre-enameloblasts and enameloblasts. Now pre-enameloblasts gain their nutrition from this area. Nuclei and Golgi complex of the epithelial cells migrate from the basal compartment towards the apex, because now the basal part of the cells are here. And this is so-called inversion of the nuclei. The cells stretch vertically and become fully differentiated enamelloblasts, which start to produce components of the enamel, bluish here, towards the layer of dentin, yellow here. These components, the organic components of the enamel, are the proteins, mainly amelogenins and enamelins. Mineralization of the matrix of the enamel occurs shortly after its secretion, which is typical for enamelogenesis. It is the beginning of the stage of histogenesis of the tooth development. Also at this stage, residual dental plate splits into discrete clusters of the epithelial cells known as the rest of Malassez. Here, this dental plate should split into discrete clusters. Differentiation stage is complete by the end of month 4 of embryonic development. Future shape of the crown now is determined, as it's shown in this diagram. Here we can see the developing of the incisor tooth. Dentinogenesis occurs with the help of the odontoblasts or dentinoblasts that differentiate from the cells at the periphery of the dental papilla. So this is the border between the future enamel and dentin. So first here was the basement membrane of the inner enamel epithelium. Just beneath the basement membrane, the mesenchyme cells now change their appearance from the typical mesenchymal cells, which contain a small amount of cytoplasm and many processes. Now they enlarge, accumulate a lot of synthesizing organelles, and differentiate into pre-odontoblasts that will in future differentiate in odontoblasts, that are the cells that produce the components of the dentin. The cells start to be connected with the junctions between the cells. They form one process apical process or Tom's fiber. And through this apical process, the cell starts to produce the organic components of the dentin that soon start to mineralize. So first, the cells are producing pre-dentin. This is non-mineralized layer of dentin that then gets mineralized and turns into dentin. This is the developed odontoblast with a well-developed rough endoplasmic reticulum and the Tom's fiber, the apical process. It is first non-branching, but soon after it gets branching, and through it the free collagen fibers and other organic molecules are secreted in the extracellular matrix. So first, the collagen fibers are located perpendicularly to the basement membrane of the enamel epithelium, but then or radially. So these are the first produced radial fibers of corf. But soon after, the apical process makes the branches that are located at different angles to the main axis of the process, and they produce the fibers that are now located more tangentially. It means in parallel to the border between the dentin and enamel. So like ameloblasts, odontoblasts are columnar cells that contain well-developed rough endoplasmic reticulum, Golgi apparatus, and other organelles associated with the synthesis of large amounts of proteins. The onset of secretory activity is marked with outgrowth of the Tom's fibers or apical processes on each odontoblast. During dentogenesis, the peripheral or mantle dentin with the radially oriented thick collagen fibers or von Korff's fibers appear first because of the orientation of the apical part of the Tom's fiber or apical process of the odontoblast. So here is the radial Korff's fibers. And here will be the mantle dentin. It is located closely to the layer of enamel. It is succeeded by the peripalpar dentin with tangential collagen fibers or von Ebner fibers. So here is the layer of peripalpar dentin. It is the most thick layer of the dentin, and in it we can find the radially oriented or parallel to the dentin enamel border collagen fibers that have name Ebner's fibers. The rapidly increasing thickness of the dentin matrix separates the bodies of odontoblasts further away from the layer of enameloblasts. So here is the layer of odontoblasts and their processes inside the dentin layer, and here are the bodies of the ameloblasts that produce enamel. While the process of dentinogenesis and enamelogenesis is going on, the thickness of both layers is increasing, and the cells, odontoblasts and enameloblasts, are now the more and more large distance from each other. So in this diagram, you can see the in the picture A, location of apical processes of odontoblasts inside the dentin, and in picture B, the collagen fibers arrangement inside the dentin. So here is the location of bodies of odontoblasts. They are located in the peripheral layer of the pulp of the tooth. Here is the layer of the dentin. This is the dentin, and this is the enamel. Here is the border between the enamel and dentin. here should be the cells producing the enameloblasts. So each odontoblast has a branchy apical process that is located inside the dentin tubules. These are the tubular cavities inside the hard extracellular matrix of the dentin, like it was for the osteocytes in the bone tissue. And some of the apical processes are somehow appear inside the layer of the enamel. They are thickened here and form so-called enamel spindles. So enamel spindles are the apical parts of the apical processes of the odontoblasts located inside the layer of enamel. In the second picture, you can see the collagen arrangement inside the dentin. Also, this is the layer of enamel. This is the layer of dentin, the border between them two, and here is the location of the pulp of the tooth. Because the form of the apical process, first portion of collagen fibers is arranged radially and form the radial or von Korff's fibers in the mantle layer of the dentin. So here will be the mantle layer. Next, when the branching of the apical process of the odontoblast is seen, now the orientation of the collagen fibers is mostly tangential. It means parallel to the border between the dentin and enamel. So here is the layer of peripulpar dentin, the most thick layer of dentin, and it includes tangential or von Ebner's fibers, collagen fibers. And the last one layer, predentin layer of the dentin. This is a still non-mineralized layer of dentin consisting of organic materials produced by the odontoblasts. The odontoblasts do not die and are still alive during all the lifespan of the adult. And the process of the dentin formation is lifelong. So the thickness of the dentin is constantly increasing. And in parallel, the volume of the pulp chamber is decreasing because of action of the odontoblasts that producing dentin towards the center of the of the tooth chamber, pulp chamber. Now about the enamel production. Enamel is produced by ameloblasts or enameloblasts with cooperation of other enamel organ cells, for example, the cells of the intermediate layer of the enamel organ. After the changing of the pole, of the polarity, now the basal pole looks towards the intermediate layer of the enamel organ. Also, this basal pole includes nucleus and the protein synthesizing organelles, including rough endoplasmic reticulum and Golgi complex. The apical pole includes the Tomes process, the apical process of the enameloblast. Major stages of amelogenesis, production of enamel, is production of matrix or secretory stage, during which the production of partially mineralized organic matrix is taking place. And the second stage, matrix maturation. Maturation of the partially mineralized enamel matrix involves cells called maturation stage ameloblast. and the maturation includes the removal of the organic material, continued influx of calcium and phosphate into the maturing enamel. The enamel is composed of enamel rods that span the full thickness of the enamel layer from the dentina-enamel junction to the surface of the enamel. Enamel rods consist of the carbonated calcium hydroxyapatite crystals. Enamel rods reveal a keyhole shape in the cross sections. This is the longitudinal section, and this is the cross section. In the cross section, they look like the keyhole shape. The head is oriented superiorly, and the tail, this part, is directed inferiorly towards the root of the tooth. Crystals are primarily oriented parallel the long axis of the rod within the head, and in the tail they are oriented more obliquely, like this. The limited space between the rods is also filled with the enamel crystals. Between the rods is inter-rod crystals. Lines of red sauce is striation observed on enamel rods that may represent evidence of rhythmic growth of enamel in the developing tooth. Enamel of an erupted tooth lacks cells and cell processes, but is not a static tissue. It can be mineralized, remineralized, or demineralized, which means a small increasing or decreasing mineral content inside the enamel. The minerals can come into the enamel from two sources. First, from the oral cavity, from the saliva of the oral cavity, and second, from the dentin, through the processes of odontoblasts and through the fluid inside the dentin tubules in which the processes are located. The enamel formation includes these stages. So these are the enameloblasts. Here is their basement membrane, the basal pole, including nucleus and the organelles, and this is the Tom's process, the apical process of the enamel. Here is the enamel dentin junction. So here should be the dentin produced by dentinoblasts. First, the columnar cells that not have the apical process product the initial aprismatic enamel layer. This layer doesn't contain enamel rods. Next, the apical part of the enameloblasts gets embedded inside this aprismatic enamel layer and thus elongates and becomes the Tom's process. So this is how the Tom's process appears. Next, the enameloblast continue to produce the organic enamel matrix and deposit it alongside Tom's processes. So the organic matrix is released through the Tom's process into its lateral directions, through the lateral surface of the Tom's process. So these depositions of the matrix are located between the Tom's processes, and they are forming the mesh of interprismatic enamel with the embedded inside them Tom's processes. So this is the scanning electronograph showing the interprismatic matrix. These holes should be soon filled with the forming enamel rods. And this is the material between the rods. So here is how the rods look like, the structural unit of the enamel, and here is the inter-enamel matrix produced by the Tomes processes, read in this picture. And next, mineralization of the matrix filling the cavities creates enamel prisms. So next, the Tomes process start to release the organic materials and the calcium salts through its apical part, just inside these cavities, and thus forming the prisms, structural and functional units of the enamel. Matrix deposition and calcification proceeds in waves, leading to appearance of redsous growth lines. And with gradual degeneration of enameloblasts and decline of the Tomes processes, the deposition is finalized by the last layer of also aprismatic enamel. So the first layer of the enamel is aprismatic, that is in touch with the dentin. And the outermost layer, first covered with the cuticle, is also aprismatic, doesn't contain prisms. But the main thickness of the enamel contains prisms and between them interprismatic material. Interprismatic enamel forms first, and then these cavities are filled with the material that is forming prisms. In this picture, you can see the ultrastructure of the ameloblast with the nucleus and protein synthesizing apparatus in its basal part and the Tomes process at its apical side. The cells are interconnected with the intercellular junctions, and their apical Tomes processes are not long, and they are constantly die from their distal parts and elongate in their proximal parts. First, they produce inter-rod material, here green in this diagram, that looks like this, and then the material of the rods, first in the central part of the rods and its peripheral part. So in the histological preparation, we can see the stage of histogenesis of the dental tissues. This is the preparation, and this is the diagram. This is the developing crown of the tooth. Crown develops first, and the root will develop just before the eruption of the tooth. Inside the developing tooth, we can see the developing dental pulp that develops from the central part of the dental papilla, consisting of the mesenchyme cells that develop into the loose connective tissue cells. Also, you can see here blood vessels that give blood supply to the odontoblasts and also pre-enameloblasts before formation of dentin and enamel. At the periphery of the dental pulp, you can see the location of the cell bodies of odontoblasts. These are the cells producing the dentin. These apical processes, Tomes fibers, go radially from the cell bodies towards the dentin enamel border. Here, the dentin here is red stained. Pre-dentin that contains non-mineralized organic material produced by odontoblast is more lightly stained. Here it is the whitish line, and this is the mineralized dentin. Here is the dentin enamel junction and the enamel layer. Now it is not so thick, but it thickens starting from the apical part of the developing crown towards the developing root. So here, bluish line is the enamel layer. Enamel is produced by the cells enameloblasts. They are located here, and in this picture here, enameloblasts that have the Toms processes at their apical sites that produces the components of enamel prisms that form the enamel. The enameloblasts contact with the intermediate enamel epithelium, then the enamel organ pulp or the stellate reticulum, and outer enamel epithelium. At the site of the apical part of the developing crown of the teeth, the outer enamel epithelium, enamel organ pulp, and the intermediate layer get more thin, come closely together, and together form the so-called cuticle of the enamel, or cuticle of the tooth, that will cover the enamel and protects the enamel during its development, but soon after the eruption of the tooth, it will be disintegrated, and the remainings of the tooth cuticle will be present just on the sides of the tooth, attaching the tooth to the epithelium of the gum. In the lower part, you can see the forming of the so-called cervical loop, which will serve to formation of the root of the tooth. The developing tooth is surrounded by the mesenchyme cells that are actually the neuromesenchyme because they are derived from the neural crest. Tooth histogenesis begins from the crown, where the dentin, enamel, and the pulp become distinguishable by the end of the embryonic month 4, whereas the root tissues, dentin, radical pulp, cementum, and periodontal ligaments emerge only by the end of month 5. The rapidly increasing thickness of the dentin and enamel separates the cell bodies of the odontoblasts and enameloblasts further away from from each other. The peripheral cells of the dental papilla differentiate into odontoblasts, the rest into cells of loose connective tissue, including fibroblasts and others. So, what happens with the structures of the enamel organ? Outer enamel epithelium in the crown of the tooth, together with the residuals of the stellate reticulum and intermediate layer, together they form the cuticle of the tooth, which is worn out shortly after eruption, except on interdental surfaces, where it attaches the tooth to the gum epithelium. In the lower part, together with outer epithelium, the inner and outer epithelium together form the cervical loop that guides the root development. Stellate reticulum provides nutrition for enameloblasts and ends in the cervical loop. Intermediate epithelium participates in calcification of the enamel and formation of the root cuticle, covering the crown of the tooth. The inner epithelium develops into enameloblasts in the tooth crown, and together with the outer epithelium form the cervical loop that guides the root development. Differentiation of the dental papilla includes the differentiation of the peripheral cells that develop into the odontoblasts and all the rest cells of the dental papilla differentiate into the loose connective tissue of the future tooth pulp. Now, about the root development. In the enamel organ, the so-called cervical loop emerges at the circular inner and outer epithelium junction. So here you can see the inner epithelium and outer epithelium junction. Here the outer enamel epithelium meets the inner enamel epithelium, and together they form a two-layered sheath, or Hertwig's epithelial root sheath, or HERS. HERS progressively grows into the mesenchyme between the dental papilla and dental sac, and participates in the dental root formation. As it grows, the inner layer of the HERS promotes the differentiation of the nearby dental papilla cells into the odontoblasts that will produce the pre-dentin that gets mineralized and turns into the root dentin, because the root of the tooth should have the dentin in its structure. After formation of the dentin in the dental root, the HERS disintegrates into the separate fragments known as epithelial cells rests of Malassez, found in the adult tooth periodontum. These are epithelial rests of Malassez. Cells of the inner layer of the dental sac, which become exposed to the dentin, and now that they can contact with the dentin, they start to differentiate into cementoblasts. Cementoblasts begin to produce organic matrix cementoid composed of collagen fibers and amorphous substance. The formation of cementum, cementogenesis, consists of two phases: phase of secretion of organic matrix and mineralization of this matrix. In the mature tooth, we have in its root the acellular cementum and cellular cementum. Acellular cementum doesn't contain cells, and cellular cementum has the cells in its structure. During mineralization, two types of cementum are formed: acellular or primary cementum, which is deposed slowly while the tooth is erupting and covers 2/3 of the root surface closer to the crown. And second type, cellular or secondary cementum, which is formed after the eruption of the tooth more closely to the apical portion of the tooth. The cellular cementum has the cementocytes in its structure, and its structure actually resembles the structure of the lamellar bone tissue, in which the cells are located inside the lacunae, connected with the canaliculi, in which the processes of cells are located. Development of the periodontal ligament is shown in this slide. Periodontal ligament is a derivative of the dental sac, mesenchymal cells of which differentiate into fibroblasts and start to produce the ground substance and pro-collagen fibers of the extracellular matrix. Periodontal fibers are deposited from both the adjoining cementum and bony alveoli and accumulate and mature with the expansion of the root and become thicker after the eruption. So periodontal ligament will help to attach the tooth to the bone socket, alveolar bone. The periodontal ligament is anchored to the root cementum and alveolar bone via Sharpey's fibers that are part of the periodontal ligament, collagen fiber bundles embedded in the cementum and alveolar bone. So one end of the Sharpey's fiber bundle is embedded into the cementum, and the second and inside the alveolar bone, where the Chypres fibers are thicker. So, sources of development of tissues of tooth are actually two: the epithelium of ectodermal source of development and the mesenchyme. Neuromesenchyme develop from the neural crest. They make the reciprocal induction. It means that epithelium acts on mesenchyme cell, make them to develop in some specialized cells, and mesenchyme in turn acts on the epithelium cells, make them develop into some other cells. Epithelium gives rise to the enamel organ that give rise to the enamel of the mature tooth. Mesenchyme give rise to the dental papilla and dental sac, and in mature teeth, dentin and pulp that develop from dental papilla, and cementum and periodontum from the dental sac. So in this diagram, these are the temporary organs in the developing tooth, and these are the parts of the mature tooth. In our histological preparation, you can see the later stage of tooth development. Actually, this is the stage of histogenesis, because you can see here the development of dentin and enamel. Here is the epithelium of the oral cavity, residual dental lamina, and the future permanent tooth is also here. This is the developing milk or baby tooth, in which you can see already developed crown and non-developed root of the tooth. The enameloblasts are producing enamel, bluish-purple line here. Odontoblasts are producing dentin, red in color here. Near the cell bodies, you can see the pre-dentin, yet non-mineralized matrix, organic matrix of the dentin that is more lightly stained. This is the developing pulp of the tooth. This is the dental papilla cells that are developing into the loose connective tissue of the future dental pulp. This is the outer enamel epithelium, pulp of the enamel organ, and here should be the intermediate layer. Those three form the cuticle of the tooth, covering the crown of the tooth. And in this part, the cervical loop is formed that guide the formation of the tissues of the developing root of the tooth. The epithelial root sheath is not included in the tissues of the root, but it guides the developing of the dentin and cementum and periodontum. Here is the mesenchyme, non-condensed, and here is the condensed mesenchyme of the dental sac that will give rise to the cementum and periodontum of the tooth. Periodontum will attach cementum to the bone tissue of the alveolar bone, which is now seen as the developing spiculae or trabeculae. At the high magnification, the central pulp, part of the pulp, and the peripheral layer of the pulp containing cell bodies of odontoblasts, pre-dentin, non-mineralized yet, and dentin, mineralized, through which the apical processes go towards the enamel-dentin junction here. Thin layer of enamel is produced by enameloblasts with their apical processes of tomes. And here is the cuticle of the tooth. Also the same in this picture, enameloblasts that are covered with the forming cuticle of the tooth. Apical parts contain the apical tomes processes that produce the components of enamel. Enamel is more basophilic. This is the enamel-dentin junction. The dentin is more eosinophilic. This is the mature mineralized dentin, and this is the pre-dentin, non-mineralized, produced by the dentinoblasts or odontoblasts, which cell bodies are located at the periphery of the dental pulp. And here is the developing dental pulp. Here the loose connective tissue is developing. The same as here, enamel produced by enameloblasts here, which have the apical tomes processes. Dentin, pre-dentin, produced by odontoblasts. Inside the dentin, you can see the radially oriented dental tubules in which the apical processes of odontoblasts are located. Also here, these radial lines inside the dentin are the location of dental tubules in which the apical processes of odontoblasts are located. So oral cavity epithelium give rise to the dental lamina, then enamel organ that finally give rise only to the enamel of the mature tooth. Also the cuticle, but it is very soon is disrupted. The neuromesenchyme from neural crest gives rise to the dental papilla and dental sac or dental follicle that give rise to the pulp and the cementum of the tooth, and also the peripheral layer of dental papilla give rise to the odontoblasts that make dentin. Here is the baby tooth development chart. So first teeth are the lower incisors, first that are appearing in the oral cavity of the baby. Baby has 10 teeth on both jaws. Here is the dentition of the baby and the permanent dentition. Babies do not have the premolars, and they have only 2 pairs of molars in each side of each jaw. Permanent tooth buds are located below the baby teeth and are starting to form as soon as the baby teeth are formed, but they are located in the jaw and wait for their time to erupt. So this is the location of the permanent teeth in the 4-year-old baby. So here is the germ of the permanent tooth that arises from the same dental lamina from which the baby tooth is developing. We are seeing in our histological preparations the developing baby tooth and also the germ of a permanent tooth. And during the teething eruption of the permanent tooth, you can see the crown of the falling milk tooth that is pushed out with the formed permanent tooth. And we have some teething theories. It is not very well understood how the teeth are erupted, why they are erupted. So first theory is the dental root growth theory, contemplates the pushing off effect between the growing dental root and the fundus of the bony alveolus. It means that, as you know, the dental root develops just before the eruption, and while it grows, it pushes out the tooth. And this explains why the tooth is erupted. The second theory, hydrostatic pressure theory, considers two possibilities. First, a critical increase in the hydrostatic pressure of the tissue fluid of the tooth root area pushes the tooth through the gum. And second, the critical increase in the pressure is caused by the formation of the dental pulp, which boosts the volume of the dental papilla in its apical portion. And this hydrostatic pressure also pushes the tooth out of the alveolus. Third theory, the theory of bone remodeling theory, considers the eruption a consequence of alternating growth and resorption at the fundi of the bony alveoli. It means that the alveoli bone tissue grows and it makes the tooth to arise just above the surface of the gum. And number 4, periodontal traction theory comes in two versions. First, the propulsive force arises from the shortening of the bundles of collagen fibers in the forming periodontum. It means that periodontum that forms at different angles between the cementum and the bone. It includes collagen fibers that can shorten and pull the tooth out of the gum. And another version, that the combined contractile activity or migration of periodontal myofibroblasts is transmitted to collagen fibers, which leads to teething. In this theory, you can see the action of myofibroblasts, that, as you know, are the cells of the fibroblastic differome that have in their structure actin and myosin microfilaments that can contract. And these myofibroblasts, as the scientists consider, they are attached with one end to the bone of alveolus and to the with the second end to the tooth, and also pull the tooth out. You should read about the teething theories and about all other information on this topic in your essential textbooks. So major information is in the essential textbook. Okay, so this is all. Thank you and goodbye.